So we start off by looking at the innate immune system. So the immune system consists of two parts, an innate immune system and an adaptive immune system. So the innate immune system is going to be the immune system components that we are born with. These are components that form the first line of defense against pathogens and all pathogens, so prevent pathogens from getting in. And then if pathogens do get in, to then of course get rid of, eradicate these pathogens. And then the adaptive immune system is also an immune system, of course, cells that we're born with. But the difference is the adaptive immune system cells adapt to their pathogens. So the innate immune system, for the most part, doesn't have a specific pathogen that these immune system components attack. The adaptive immune system is the immune system that attacks specific pathogens, right? So there's binding that has to occur to specific pathogens or specific components on pathogens. So that's one difference. The other is the adaptive immune system. Once it does bind and attack it, or comes in contact with a specific pathogen, the adaptive immune system cells adapt to that by proliferating. So that one adaptive immune system cell, when it comes across its pathogen, there's an interaction that occurs, and now you have more and more of those cells. Okay? So that it means that the immune system has adapted. It's adapted to having encountered its pathogen. While the innate immune system will do the same thing. It binds, right? It binds, not specifically. There are some specifics, but not as specific as the adaptive immune system. So what that means is, for example, one of the cells that's part of the innate immune system is a neutrophil. So neutrophil, for the most part, will attack bacteria. So you may say, well, isn't that specific? But it's not a specific bacteria. It's just bacteria in general. Okay, so that kind of lowered specificity, if you will. So that neutrophil comes in contact with a bacterium. It'll take it in and it'll destroy it. But what won't happen is that neutrophil won't then divide, and now you have more of those same neutrophils. Okay, so that's the difference. So we start off by looking at the innate immune system. Also, you should know that the innate immune system and the adaptive immune system both interact with each other, right? So the innate immune system will stimulate the adaptive immune system, and then the adaptive immune system will then talk or stimulate the innate immune system. Okay, so they interact with each other. So we start off by looking at the innate immune system. So as I said earlier, the innate immune system is the first line of defense, right? So the first line of defense, so here's obviously us. And so the first line of defense that you see here is going to be the external components: skin, mucous membranes, antimicrobial substances that are present in skin and mucous membranes. The second line of defense, so this is keeping the skin, keeping the pathogens out. If the pathogen were to get in, which of course we know that they do, the second line of defense is going to be inflammation, localized inflammation, localized edema to prevent the pathogen from getting into the circulatory system and then having access to the whole body. Fever, of course, because high temperatures should kill the pathogen. Of course, if it's a pathogen that can handle high temperatures, it can only help it, right? And phagocytes. So phagocytes, so cyte means cell. Phagocyte is a cell that goes through a process called phagocytosis. Phagocytosis is just simply endocytosis, where the cell takes in something from outside, right? And so that is endocytosis. But phagocytosis is bringing in that component into the cell and then destroying it. Okay? So phagocytes are cells that go through the process of phagocytosis, which is bringing in a foreign particle that the cell detects. Could be a pathogen, could be cellular debris, and it brings it in, keeps it in a vesicle, destroys it, and then releases it. So this is how then you get rid of pathogens. And then the third defense would be something called humoral and cellular immunity. So humoral and cellular immunity is part of the adaptive immune system. So these out here are part of the the innate immune system, and then humoral and cellular immunity is part of the adaptive immune system. And we'll learn more about this in the next chapter. So these are the cells that are part of these two immune systems. So part of the immune system, or innate immune system rather, are cells called basophil, eosinophils, mast cells, and then you have cells that are kind of in between. I wouldn't say that a neutrophil belongs in the adaptive immune system, but they kind of have this role that they interact between the innate. So these cells allow interaction between the innate and the adaptive immune system. So those are then your neutrophils, monocytes, dendritic cells, and the natural killer cells. And then part of the adaptive immune system are going to be your B cells and T cells. So the immune system cells are called white blood cells, right? Or leukocytes, more specifically, leukocytes. So when a person is ill, and what they'll do is, if you want to know, is there an infection, you do a white blood cell count. And if you have a high white blood cell count, it can indicate an infection, depending on which type of cell. So since neutrophils are cells that specifically attack bacteria, then you would have an uptick in white blood cells, in neutrophils specifically, right? So it could indicate bacterial infections, it can indicate autoimmune diseases, or even side effects from medications. Now, low white blood cell counts can indicate viral infections. So why do you think that is? Why is that? Why would the low white blood cell count indicate viral infections?
It's because isn't the invasion attacking the white blood cells as well? So like AIDS attacks white blood cells.
Yeah. So it can. It doesn't always. It can. Depends. Different viruses preferentially infect different types of cells. So if you have a low white blood cell count, that means something's happening to the white blood cells, right? And if something's happening to the white blood cells, mostly it's going to be a viral infection. So good, Brian. Pneumonia, autoimmune diseases, because it could be affecting the white blood cells itself, and also cancers, because it could be just affecting the white blood cells. Okay. So the normal percentages of these white blood cells would be indicated as you see here. So the most abundant type of white blood cells we have are neutrophils. So about 60 to 70% of the WBC counts are neutrophils. We have monocytes, and then the lowest is going to be our basophils. You already know from 202 blood lecture that blood consists of two components, right? You have the formed element component of blood. This is going to be comprised of all the cells, white blood cells, leukocytes, platelets, and then you have the matrix of blood, which is of course plasma, which is just the water and all water soluble components within plasma. So antibodies, which we'll see what they are coming up, would be present in the plasma. So let's go on to then take a look at the differences between innate and adaptive immune system, which kind of already started, and we'll look at what toll-like receptors are. Okay. So the immune system's job is to prevent pathogens from getting into the cell, from colonizing either on our body or in our body, to clear cellular debris. So you would think, where's cellular debris coming from? But cellular debris can come from when our cells are dividing. When their cells are dividing, you have cytoplasm that's left over from the divisions and things like that. Also, immune system helps in clear proteins that have been misfolded, and or cells that are dying themselves just from old age and things like that. So the immune system is basically, housekeeping, right? They want to make sure that things that aren't going to pile up, if you will. So immunity. So this is some terminology. Immunity is the ability to fight a disease. This immunity is going to be afforded by the presence of these immune system cells. Susceptibility is so such a weird wording, but this is from the textbook. But susceptibility just means that you're susceptible to the disease. Something's happening. Either you don't have enough white blood cells, or you don't have any, as in the case of AIDS, as Ryan was speaking of, or HIV rather. Innate immunity, this is the defenses against pathogens, but these are defenses that are present from birth. These are defenses that don't adapt to the encountering of their pathogens, as opposed to adaptive immunity. This is immunity that is specific towards a specific pathogen. It is a slower response, and we'll see why coming up. Innate immune system response is faster. And there is a memory component. What this means is that the cells of the adaptive immune system, when they come in contact with their specific pathogen, they're going to go through division, cell division, proliferation, and create many more of the same type of immune cell, so that the next time your body encounters that same pathogen, you already have these immune cells to attack. And so the reaction to a second or third tertiary infection, the reaction rate is going to be faster. Again, just some terminology. So toll-like receptors, these are receptors on immune cells that attach to components on pathogens. Proteins or glycolipids that stick out on pathogens, that is what these toll-like receptors detect and then bind to, right? So there are different types of, or they're toll-like receptors that have different, so for the different types of proteins or different types of glycolipids that are sticking out from a pathogen, you will have one specific receptor for these different permutations of these proteins, right? So toll-like receptors are on host cells, which is mostly the immune cells. And these receptors, what they do is they attach to pathogen-associated molecular patterns, so PAMPs. Okay. So PAMPs are going to be just what it says there. These are molecules that are sticking out from the cells that these toll-like receptors bind to. When you have this binding, what happens is you have the release of chemicals called cytokines. So cytokines are chemicals that cause growth, that cause differentiation, right? So when these cytokines are released from that host cell that has the toll-like receptor, you're going to have a reaction that causes division, increased division of that cell. So then now you have more cells that have that same toll-like receptor. Okay, so then if you come across that same PAMP again, you'll have a faster reaction. So we go on to now looking at the first line of defenses. So the first line of defense, of course, is skin and mucous membranes. So let's see how skin and the mucous membranes help prevent infections. So the skin, you know already from 202, it's got two parts to it, a dermal layer and then, the outermost layer, which is the epidermis, and then the layer that is in deep to the epidermis is the dermis layer. The epidermis, so epidermis is made up of 5 layers, 4 layers, 5 if you have, when you're talking about thick skin, but 4 layers. And the outermost layer is made up of 30 to 40 stratified squamous cells that are keratinized. Keratinized means that these cells have a protein called keratin deposited in them. And what keratin is are these fibrous proteins. And once they get deposited in cells, the cell actually just dies. So on the very top layer of the epidermis, the stratum corneum, you have 30 to 40 layers of these dead keratinized stratified squamous cells. So that keratinization, what it does is you create, so they're stratified, so there are multiple layers. So that in itself is a physical barrier. The second thing is you also have glycolipids that are present, so that prevents, it creates a waterproofing, if you will. So it prevents water-soluble pathogens or water-soluble molecules from getting in, right? So it produces this protective sheathing. Shedding and dryness of skin also inhibits microbial growth. So again, the very top layer of the epidermis consists of 30 to 40 layers of dead cells. So we're always sloughing off the topmost layer of our epidermis. And so whatever pathogen was there is, of course, going to be released off of, along with those cells that are sloughing off. Okay. So this is what I was talking about. This is going to be this right here, from here to here. Right, so just like it says here, from here to here is the dermis layer, and then you have, excuse me, epidermis layer, and this is dermis. And this top layer here, this is the 30 to 40 layers of dead skin, and this is, of course, creating that protective barrier. Then you have mucous membranes, right? So mucous membranes are epithelial layer of cells, the cells made up from the epithelial tissue group. Mucous membrane is the part outside the tissue or around the inner cavity. And you have these cells that are present called goblet cells. Goblet cells are modified epithelial cells that produce a chemical called mucus, right? So membranes that have goblet cells are called mucous membranes, right? So it's an epithelial layer that lines the GI tract, the respiratory tract, the genitourinary tract, right? So this is going to be a layer that is present anywhere we have interaction with the external environment. And these cells called goblet cells are cells that produce mucus. Mucus is a viscous, thick liquid. And what it will do is it'll trap dust, it'll trap pathogens, and then be able to reroute so that mucus moves to regions where we can then get it out the body, right? So if it's in the esophagus, then of course it's going to trap things, and then it'll go to the stomach. You have lots of hydrochloric acid there, and it gets destroyed. If it is in the respiratory, what will happen is the mucus will trap dust or pathogens that you breathe in, and it'll trap it, and then it moves it into the esophagus, and then it gets destroyed as it gets into the stomach, right? So mucus is a viscous chemical made up of glycoproteins. And so, as I said, it traps microbes and prevents the tracts, of course, from drying out. So in our eyes, we have, so it's called a lacrimal apparatus. So what it does, lacrimal meaning pertaining to tears, right? So it produces tears. So tear ducts release tears. The lacrimal apparatus drains the tears and then washes the eyes. And within the tears, you have chemicals that will prevent colonization of bacteria and yeast and so forth, right? So you don't need to know this. This is more 202, but I just wanted to show you where it is. So what are some of the physical factors that allow for this first line of defense? So you have, again, as I said earlier, mucus is present, traps these pathogens or cellular debris, and then that mucus is moved to areas, moved to places where then you can get rid of that mucus. So that moving that mucus is done via ciliary escalator or ciliary movement, right? Transport the microbes trapped in mucus away from the lungs. Earwax, of course, prevents microbes from entering the ears. Urine, of course. Yes, urine cleans the urethra, which is the tube that comes out of, that exits the urinary bladder, and it allows for urine to exit the body through, but that's not the function of urine. I would say more really the pH of urine is what is, it has the immune aspect to it. So pH of urine is about, normal pH of urine is like 6.0, so that's kind of lower than physiological pH, and that's what's preventing microbial growth within the urethra. The function of urine is, of course, to um, release metabolic byproducts from the body. Vaginal secretions, also same thing, like along with the ciliary escalator or the mucus. So moves microorganisms out of the vaginal tract. Peristalsis, defecation, vomiting, diarrhea, these are all going to be, well, vomiting and diarrhea specifically are um, responses to the presence of pathogens. So vomiting and diarrhea is a response, a specific response to the presence of pathogens. Peristalsis is just simply movement of the smooth muscles along the GI tract that causes movement of components throughout the GI tract. Defecation, of course, is just necessary to remove undigested material. So peristalsis and defecation isn't really part of the immune system. I don't know why. I mean, does it have a role? Yeah, it has a role, but that's not their main function. The main function of vomiting and diarrhea is to get rid of pathogens. So vomiting and diarrhea definitely have an immune function, whereas peristalsis and defecation have secondary roles as immune system functions. So those are physical factors. The chemical factors that are present are going to be sebum. So sebum is present in the exocrine glands that are present in skin. So sebaceous glands and apocrine glands. These are glands. This is what material you would have learned in 201. So sebum is present, and sebum is sweat, which is like 99% water and salt and antibacterial, or not really antibacterial, but chemicals that prevent bacterial colonization, yeast colonization, and also it has a low pH. So the pH is about 3 to 5. So it's actually called an acid mantle. So on our skin, in addition to the keratinized squamous, stratified squamous cells, we also have the presence of sweat and also sebum, and these are, and also along with that, you have lowered pH. So on our skin has a low pH, and so that prevents bacterial and yeast colonization. Lysozymes, excuse me. These are going to be present in sweat, in tears, saliva, and urine. And what these are going to do, so these are enzymes that destroy, that hydrolyze proteins, so they can destroy bacteria cell walls. Now, do you think these would affect gram-negative bacteria as much?
No, because of the two layers.
Yeah, because the outer layer is with gram-negative is going to be our membrane. So gram-negative is a specific bacteria that really causes most of our problems. Gram-positive are much easier to attack. Gastrojuices, so gastric secretions. So these are secretions within the stomach and throughout the small and large intestine. So within the stomach, the secretions are very acidic. Hydrochloric acid, pH is 1.2 to 3.0, so very low. And then pH in the intestines are higher pH, about 8 to 9 pH. So that also prevents colonization of bacteria and yeast. Vaginal secretions are also going to be low pH, and so that also prevents colonization. So we learned about the microbiome in the very first class. We talked about how we have these specific microbiomes, specific bacteria, specific other types of microbiomes, or microbes rather, like yeast, but for the most part, bacteria, right? We have this, each one of us has a specific bacterial or microbial collection, right? And so that is our normal microbiota. That's what normal microbiota is. This is our normal microbiome. Now, if you think about the microbiome world, all these organisms, they're always fighting with each other. They're constantly trying to kill each other off because they're all fighting for resources, right? So if they had a chance, they would definitely kill each other. And so how do they prevent? How does a yeast prevent a bacteria from infecting it or killing it? It'll produce chemicals that's going to counter whatever it is toxin that the bacteria is throwing at it. That's how, listen, we humans have not created any kind of I guess innovations of our own. We've just copied what the microbes do. Okay, so penicillin, of course, comes from penicillium, the mold that created that chemical. It was just accidentally observed, right? So these microbes are constantly fighting with each other, and our microbiome, then of course the question is, well, how come all these microbes that we have aren't dead? Because of this reason, because they can find ways to counter these, spy versus spy kind of thing. They counter these different attacks that they encounter, and then they themselves launch attacks. You see what I'm saying? So they kind of are in this battle, and then they come to this kind of equilibrium where you can't fight them all off, right? And you can't kill them all off either. So you kind of settle at this equilibrium where the number of cells living is equal to the number of cells dying, right? In all the populations. So the normal microbiota compete with pathogens that enter. What does that mean? So you have your normal microbiota, right? Normal microbiome. And these are, as I said, microbes that are duking it out with each other. Now you've got another pathogen that enters. You've eaten it, you swallow it, you've breathed it in, it's gotten into you, however it got in. So now they detected this pathogen, and now they will then, of course, try to kill off that pathogen. So our normal microbiome actually helps us fight off other pathogens. Now, you may say, well, how come our microbiome doesn't infect us or doesn't cause us problems? Because they're busy duking it out with each other, right? If you were to change the balance in that normal microbiome, it can become a problem, because now the unchecked microbe, now it has time to then affect our cells. Does that make sense what I'm saying?
Yes, Professor.
Right? So our normal microbiome, it's not that these are altruistic microbes and they're not going to attack our cells. Yes, they will if given the chance, but they're not given the chance because they're all fighting with each other. Now you add another pathogen, and now you'll have an extra layer, right? So you have an extra fight, if you will. Sometimes what happens is that pathogen can get rid of a certain population of microbiomes. Okay, then what you have is that that pathogen that killed off certain microbes, right? These microbes that would have kept the others in check that are now gone leaves the other microbes that you have in your microbiome unchecked. That becomes an issue then, right? That's when you have infections. You see, so this is all about the balance of power. It's all about the balance of power. Okay, so normal microbiotas compete with pathogens via something called microbial antagonism, which is exactly what I've explained. So what they do is, again, so these microbes will produce substances that are harmful to the pathogens and harmful to other microbes also. And they alter the conditions that affect the pathogen survival, right? So they can produce and release acidic secretions and what have you. So they kind of keep each other in check. Okay, so this is how the normal microbiome is also part of our immune system. Then we have commensalism, which we've already learned about. This is when one organism benefits while the host is unharmed. So for the most part, our microbiome is in a commensalist there's a relationship with us, except for some bacteria that also will further digest vitamin K, or not digest, excuse me, metabolize and produce the functional form of vitamin K within our body. So in the large intestine. So in the large intestine, you don't have any more digestion that human cells conduct. The digestion that takes place in the large intestine is because of your microbiome. But for the most part, our microbiomes, we're not affected directly, right? But we are indirectly, right? Because they do help us, as we just established, okay? Probiotics, these are chemicals so these are live microbial cultures that we take in to help boost our normal microbiome. So, things like yogurt and things like that. Recently, there's kombucha, right? I don't know. I personally would not trust kombucha. I don't know what kind of live microbial microbes are in there, and I don't know how that would affect, the different microbiomes. But for the most part, I'm sure it's all balanced. So that's the first line of defense, microbiome, skin, mucous membranes, and pH secretions, and so forth. Then the second line of defense is going to consist of cells, right? So this is when the pathogen's already in and it hasn't been killed off by these different components that we talked about. This is when our cells then of the immune system have to come into play. So leukocyte functions, and then we'll look at how these leukocytes, what they are and how they do what they do. So as I said earlier, remember with blood, there are two components to blood. There's the form elements part of blood, and then there's the matrix of blood, which is plasma, which is the liquid portion. So cells and cell fragments that are suspended, that are in blood, so the cellular components of blood are called formed elements. And so those cells are erythrocytes, leukocytes, and platelets. These are cells that are all created within the bone marrow via a process called hematopoiesis. So you don't need to know slide 22. This is all 202 material. You don't need to know for this class how these cells come about to be. But just in general, you have stem cells. Always, everything starts with stem cells. Stem cells are going to differentiate into two other more differentiated stem cells. And then those stem cells are going to give rise to these different immune system cells. Now, notice that the two main stem cells are your myeloid stem cells and the lymphoid stem cells. So the myeloid stem cells give rise to these white blood cells here. Okay. So the white blood cells are divided into two major groups, granulocytes and agranulocytes. Granulocytes are cells that have visible granules. If you look at them under the microscopes, you see visible granules in the cytosol. And then agranulocytes are cells, if you look at them under the microscope, you don't see the granules. It's not that they're not there, it's just that they're not easily visible. The cells that belong to the granulocytes are, I remember it as NEB because it's just easy. So that's neutrophil, basophil, or neutrophil, eosinophils, and basophils, so NEB. And then the cells that belong to the agranulocytes are going to be the monocytes, T cells, B cells, and the natural killer cells. So again, granulocytes, these are the leukocytes that have visible granules in their cytoplasm. The granules within them are going to have different types of different components that will help get rid of pathogens. So the cells that belong in the granulocyte group are neutrophils, eosinophils, and basophils. So neutrophils are cells that are phagocytic, so they can extend their cytoplasm, bring in the pathogen, keep it in the vesicle, and then destroy it. So neutrophils are phagocytic. their main role. So these are, remember earlier I said neutrophils are the most abundant type of cells. So these are the first soldiers on the scene, if you will. So they work early in the stages of infection. Eosinophils, these are also phagocytic, but for the most part, what they do is they release hydrolytic enzymes, and therefore they're going to digest whatever is outside of that eosinophil. Neutrophils bring in the pathogen and destroy eosinophils, release hydrolytic enzymes and destroy the pathogen that's outside of the cell. So can they go through phagocytosis? Yes, but unlike the neutrophils, they can also release these enzymes. So eosinophils are specifically they specifically target parasites and helminths. So helminths is worms. So they target worms, so worm larvae and so forth, because these are too large to endocytize, so they have to be destroyed outside of the cells. And then you have basophils. So basophils, when they are activated, they release a chemical histamine. So histamine is a very strong vasodilator. So when histamine is present, capillaries become very permeable, and water is going to rush out of the capillary, and then you will have localized edema, right? So that's why basophils have a role in allergic responses. When you think about allergic responses, you think of hives and swelling, and this is because of basophils causing increased permeability in capillaries. Overaction or overactivation of basophils can lead to anaphylactic shocks, which is, of course, deadly, can be. Okay. So this is what neutrophils look like. Neutrophils have this kind of multi-lobe nucleus. It kind of looks like it's multiple nuclei, but it's just one nucleus. It just has multiple lobes. Basophils are going to be small, dark cells when you look at it under the microscope, and it just looks like a dark dot or spot, because you can't see the nucleus because it's filled with granules. And then eosinophils, and you can tell the difference between a neutrophil and an eosinophil is by the nucleus. With the basophil, the nucleus kind of has this C-shaped or U-shaped nucleus. With the neutrophils, you have this multi-lobed nucleus. Now we talk about agranulocytes. So agranulocytes are the leukocytes that they're not visible. The granules are not visible, hence agranulocytes. The cells that are agranulocytes are monocytes and lymphocytes, and then dendritic cells. So lymphocytes are the T cells, the B cells, and the natural killer cells. Okay? These are part of the adaptive immune system. Dendritic cells, these are cells that are found within the skin, the mucous membranes, and the thymus, and they are phagocytic. Dendritic cells, however, are part of the innate immune system. They're not part of the adaptive immune system. Then you have monocytes. So monocytes are cells that are present in blood, and when there's a pathogen present, that monocyte leaves the capillary, and once it leaves the capillary, it goes through a differentiation process and becomes a macrophage. A macrophage is a phagocytic cell, but they're very large, and they can phagocytize large number of pathogens or large amounts of cellular debris. That's kind of the difference between neutrophils and macrophages. Although, well, that's enough for you to know that. So monocytes mature into macrophages when they come in contact with their pathogen. And so now so then you have these macrophages. Now, there are two different kinds of macrophages. There are monocytes. So macrophages can stay within the dermis region of the skin. So these are called stationary. And then you have macrophages that continue to stay in blood and travel through blood. Okay? But their main function is phagocytosis. Okay, so this is a good slide. It tells you what these different cells can look like. So here is a monocyte, right? And it has differentiated into a macrophage. This is a dendritic cell. You can tell it's a dendritic cell because it has these little, this is not such a good slide, but they would have lots of these arms sticking out, or branches. That's why they're called dendritic cells. The lymphocytes, there are three types: the natural killer cells, T cells, and then B cells. You don't need to know what they look like. I'm not going to put pictures of this for you to know. You would need to know what their functions are, but we're going to go through the functions one by one. Specifically for these three. Dendritic cells, what they do is they go through phagocytosis and they interact with the adaptive immune system. Monocytes, they go through the process of phagocytosis. This is when, so it's not the monocyte that can be phagocytic, it's the macrophage. Okay, so it has to differentiate first. Okay. Okay, you don't need to know this part. So 28, 29, 30, 31, you can skip. Let's go on to learn about phagocytes. So phagocytes are cells that go through the process of phagocytosis, right? So they go through the process of phagocytosis. They also interact with the adaptive immune system. Okay? So let's see how they do that. So again, phagocyte means a cell. So cyte means cell. Phago in Greek means to eat. So it goes through the process of phagocytosis, brings in something from the outside of the cell and then destroys it. As I was talking about macrophages earlier, right? I said they're fixed macrophages and free or wandering macrophages. So once the monocyte differentiates into a macrophage, that macrophage can either stay within the blood and continue to circulate, or it can leave the capillaries and then stay within the organs and tissues or within the dermis region. So fixed macrophages remain within tissues and organs. So if a pathogen is present, then you don't have to wait for monocytes to leave the blood cell or the circulatory system. They're already there. And then you have free or wandering macrophages, and these are they are present in blood and they travel around the body. So this is a good slide. It's showing you a macrophage that is endocytizing or phagocytizing rather bacterium that you see here, right? So these are your bacterium, rod-shaped bacteria that are getting phagocytized by this macrophage. So I keep talking about this process of phagocytosis. So this is phagocytosis. So phagocytosis has these several steps. And the steps start off with step one, something called chemotaxis, and then followed by adherence. So chemotaxis is this, right? Taxi, you could imagine what it means is to move something, right? Transport something, right? So chemotaxis is movement of a phagocyte either towards something or away from something. So when a cell moves towards something, it's called positive chemotaxis. And when a cell moves away from something, it's called negative chemotaxis. So in both instances, moving towards something or away from something is because of the presence of chemicals, right? So chemotaxis, it involves chemical signals that, or positive chemotaxis more exactly, is when chemicals attract phagocytes to a certain region. So you have these phagocytes that are in the blood and they exit. Well, how do they know which way to go, right? It's not like they can see the bacterium or the pathogens. So when bacteria or pathogens enter the skin, a few things happens. So the reason why something entered is either you got cut or you ingested or breathed it in. So these pathogens secrete chemicals. These are chemicals that phagocytes can then detect, and then they go towards them. So first you have the release of these chemicals and you have the movement of phagocytes towards the microorganisms. The second thing that happens is the attachment of the phagocytes to the surface of the microorganism. In other words, the phagocyte attaches to that microbe. Then what it's going to do is it's going to go through the process of ingestion. So the phagocyte coats that pathogen with a chemical, making the, ingesting that pathogen easier. That process is called opsonization, right? It kind of has the word, well, it reminds me of optimization. So making it better process or easier, more effective process of digesting these pathogens. So opsonization, and then finally is digestion, right? So where the microorganism is digested or broken down inside of that phagocyte. Now, when that phagocyte brings that pathogen in, it keeps it within a vesicle. Otherwise, the pathogen is free inside the cell and it'll kill the cell, right? Its toxins will. So in order to prevent that, of course, the phagocyte brings in the pathogen, keeps it within a vesicle. That vesicle then fuses with another vesicle in the cell called the lysosome, and those two vesicles fuse, and then that pathogen inside is exposed to those enzymes of the lysosome and it gets destroyed. So chemotaxis of that of this white blood cell towards that pathogen, it attaches to that microbe, right? So here's the attachment process. Remember earlier we said the toll-like receptor. It's a receptor on the phagocyte that detects the certain types of molecules on the surface of microbes. So this is our PEMP. This is in this case, this cell is detecting the peptidoglycans that are of the cell wall of the bacteria. So it'll attach to it. So that's the adherence part. What it's going to do is then it's going to ingest it. So the cytoplasm kind of just envelopes that pathogen, and then what it does is it encapsulates that pathogen within a vesicle. That vesicle is called a phagosome. Some means body. This is a phagosome. That phagosome then will fuse with the lysosome. As I said, a lysosome is a vesicle within our cells that have hydrolytic enzymes. When that phagosome fuses with the lysosome, it's called a phagolysosome. But so what happens is now that microbe is exposed to those hydrolytic enzymes, they get destroyed, and that's the digestive process. The sixth part then, of course, is to extrude or get rid of those pieces. They're no longer dangerous, and it just enters into blood. From blood, it's going to then circulate to the kidneys, gets filtered out, or gets circulated to the liver first, gets maybe detoxified. From there, enters kidneys, and then, of course, it's released via urine, or if it's happening within the digestive system, it'll be part of the fecal matter. So this is phagocytosis, and I go through it in my lectures that I worked out on the board also. So there are different types of so these are the different types of microbes that we encounter, and these are some of their factors that make them problematic to us. So Streptococcus pyogenes and Streptococcus pneumoniae, on their cell surfaces, what they have are M proteins. So M proteins make it difficult for our white blood cells or the phagocytes to bind. They are also capsulated, so that also makes it difficult to bind to them, adhere to them. Staphylococcus aureus. So on our skin, we have Staphylococcus aureus, but on our skin, it's not a problem, but once it gets in to our body, then it becomes a problem. So what they do is they actually get into the phagocytes. So the phagocytes phagocytose them But then what they do is they kill the phagocytes, right? They have leukocidins. Um, so that's why they're pathogenic. Listeria monocytogenes, cytogenous rather. So this is what creates mono, right? What they do is they lyse phagocytes. So these are cells, these are bacteria that are dangerous to phagocytes because, well, they lyse phagocytes. Shigella or rickettsia, these phagosomes or phagocytes have can't really either detect them as well, or they're unable to phagocytize them or have a difficulty in doing so. HIV, which is of course a virus, Mycobacterium tuberculosis. These are going to prevent the lysosome fusion. So these, specifically with HIV, it's a virus. It gets in. Specifically, it will target T cells, T helper cells, and then they're going to kill the T helper cells. But what these do is they prevent, they if they do get phagocytized, so a virus rarely gets phagocytized because it sends its genetic information or into our cells, so we rarely even detect it. But Mycobacterium tuberculosis, what it does is it prevents that fusion of the lysosome. So now it's hanging out within the phagosome in its little vesicle, and it can just then get released from there and then cause issues. Right? Coxella burnett's, these actually survive in high. So the hydrolytic enzymes, what they are, these are enzymes that are at very low pH. That's how they destroy these microbes that are within the phagolysosome. But what C. burnett's, they thrive in very low pH. So they survive within the phagolysosome. Right? So these are the microbial evasions of phagocytosis. This is how these microbes, which we encounter, counter the phagocytes. So let's talk about inflammation, right? Because inflammation is the second tier of defense. So what are the 4 signs of inflammation? Obviously, redness, swelling, localized edema, pain, and it's hot when you touch it. The functions for inflammation is to, one, because of the heat that's produced and destroy, and also, well, you have white blood cells that are present there, right? Inflammation means you've made the capillary more permeable, so you have more fluid that exits. So of course, there are going to be white blood cells that come out also. Right, so inflammation also will destroy pathogens that are present, or of course limit their access to the rest of the body because it's localized in that region. Inflammation also gives the body time, because if you cut off that region that was infected, gives time to clear that debris out, dead cells, pathogens, and gives the body time to repair and replace damaged tissue. So what inflammation also does is, well, inflammation is caused by the release of chemicals that cause vasodilation, right? Which increases permeability of blood vessels. The chemicals that cause inflammation, one are histamine, which we saw is released by the basophils. They're also released by cells called mast cells. Kinin, prostaglandins, leukotrienes, cytokines. These are also going to be molecules that cause inflammation. Okay. So this is just giving you a visual of everything that I just talked about. So what we're talking about here is two processes, or the process of marginization. So here is our red, it's a capillary, and this is the tissue outside, and this is a bacterium. These, well, that's a macrophage. These small, of course, rod-shaped are the bacterium. Now, what this is describing is how the white blood cells exit the capillary. So here is our monocyte, right? Here's the capillary, and this is, of course, blood within the capillary, and these are white blood cells within the capillary. When you have bacteria outside, what's going to happen is the monocyte that's traveling along, it kind of sticks to, so there are proteins that get expressed by the within these cells on the interior portion of that capillary, and the monocyte, blood is rushing along, right? So you've got to stop these monocytes, otherwise they're just going to leave that area. So what happens is you have this process called marginization, or margination rather. And this is where the phagocytes stick onto the walls of the capillary. And that happens because there are proteins that are expressed, and the proteins that are on the outside of the monocyte sticks to these proteins. And then so it kind of sticks to the walls, the inner walls of the capillary. Then what happens is you have an increase of permeability of that capillary, and then that monocyte kind of squeezes through, like amoeboid motion, squeezes out between the cells that are making the capillary. That process is called diapedesis. This is where the phagocytes squeeze and move through in between the cells of the capillary. And then once it's out, you have chemotaxis. What's going to happen then is the monocyte, this is the monocyte, differentiates into a macrophage, and then it's going to endocytize that bacteria. So then you have phagocytosis, rather, not endocytize, phagocytosis of that bacteria. So when you do have damage because of physical trauma, or because of chemical trauma, or because of the presence of pathogens, tissue repair can't be completed, right, until all of the harmful substances have been removed and neutralized, right? So if you have a wound that's not healing, and the reason for that is because that area hasn't been cleared of either whatever agent that has caused the damage, or their agents that are still causing damage, right? So the only way a tissue can completely heal, right, is if once you remove these damaging either microbes or chemicals that are present. So what happens is at very first, once you have damage to tissue, you have clearance of the debris, clearance of the pathogens. Once that's done, then what you have is a creation of something called stroma. So this is connective tissue that starts forming fibers, connective tissue fibers, reticular fibers, elastin fibers, so forth. These start forming this supportive tissue. And then you have the division of whatever cells of that tissue were present, if it's skin, if it's liver, what have you, right? Then you have filling in of those missing cells. Okay. So this is just showing you this process of inflammation. I don't know if you could skip 46, it doesn't really make sense. So let's talk about fever. Right, we're talking about the second line of defense. Okay, so fever, phagocytes, and inflammation. Right. So let's look at the effects of fever. So fever is when you have an abnormal high blood pressure, blood temperatures. So our body temperature is normally kept at 37 degrees centigrade. The hypothalamus maintains this temperature. When we have an infection, there's an interaction between the innate immune system and the adaptive immune system. That interaction is caused by the release of these chemicals called cytokines. Cytokines cause co-stimulation, causes stimulation of, or causes interaction between the two components. And then the adaptive immune system does, because of the presence of these cytokines, will start multiplying. And another function of those cytokines is it will travel to the hypothalamus, and it causes the hypothalamus to release chemicals called prostaglandins. What these chemicals do is they reset the hypothalamus's normal temperature to a higher temperature, right? So the question has to be, right, the body doesn't just do things, right? Everything is kept in check by the endocrine and the nervous system, right? So the hypothalamus is what sets this body temperature at its normal range. If your body were to get hot, right, what would be the response? Well, you start sweating and your body starts bringing down the body temperature. But why does fever exist? Why doesn't the body prevent the high temperature? Or how does the body create this high temperature? Well, one, what happens is the hypothalamus, the normal body temperature is reset to a higher normal body temperature. Well, not normal, but a higher temperature. So that when the body increases the body temperature, you won't have these counter actions to bring that back, because otherwise that doesn't make sense, right? You bring up the temperature and then the body brings it back down again. So cytokines are going to travel. So it's pretty smart, right? The same chemical that's released because of an infection is the same chemical that resets the range of normal body temperature within the hypothalamus. What the body does is it constricts blood vessels, and shivering occurs, right? And shivering occurs to increase the temperature. So how are you increasing body temperature? By shivering. Also because you have thyroid hormone release. Thyroid hormone increases body temperature also. Then, so that is the increase in body temperature, that's fever. So once the infection has kind of subsided or is under control, then this is when the body temperature starts falling. This is called crisis mode. This is when now you have vasodilation. So once you have vasodilation, you can have sweating, and the body is going to then bring it down. You know, when you say like the fever broke, that's that part where now the body temperature is coming back to normal. And that's a good thing, because it's a good sign, rather, because it says that the infection is under control. So usually when people have fevers, the first thing at least how I grew up, first thing is to try to bring the temperature down. So is that a good thing to do you think?
Within reason, you don't want to let the person overheat to a certain extent, but controlling the fever is important.
Yeah, for sure, but not to just cool down the body right away.
Yeah.
Right, which is what I think a lot of old school was. That as soon as you have, you put like ice packs on your forehead or what have you, and try to bring the temperature back down. You just want to keep it under control, right? So this is asking, why does a chill indicate a fever? So this is a good question. Why does a chill indicate that a fever is about to occur?
Is it because it changed the threshold? So your current temperature now feels cold to you?
Good job. Yeah, absolutely. Because now the body's like, whoa, you should be at 101, not 98. So that's why you feel cold, right? Cell perception. It's pretty smart. Mind play tricks on you. So let's talk about antimicrobial substances, except let's take a break. Y'all want a break? It's 7 o'clock. I need a break. Let's put it that way. That's a lot of talking. I will see you at 7:30. Okay?
Sounds good, Professor.
That was good.
And I'll pause the recording. Okay. So far we looked at we were looking at the different lines of defenses of the immune system offers to protect us from pathogens. We looked at the outermost layers, and those are your mucous membrane, skin, and so forth. And then we looked at the second line of defense, which is then the phagocytes, fever, phagocytes and fever to help prevent further pathogen and inflammation, to prevent further pathogen colonization, right? We're keeping it with that. We're now going to look at antimicrobial substances that our body produces. Now, before we move into the complement system, I want to give you some. So let me move this out of the way. The complement system consists of a group of about 30 or 40 proteins called complement proteins. And they're called complement proteins because they work in tandem with each other. They complement each other. Okay? Now, these complement proteins are produced by the liver. So they're serum proteins produced by the liver that helps the immune system in destroying microbes, in identifying and destroying microbes. And they work, as I said, they work in tandem with each other, these group of complement proteins, and they create this kind of biochemical reactions. It's called a complement activation. So it's this cascade of events that occurs because of the interactions of these different proteins. Now, all of this starts off with when an antibody binds to its antigen. So let me put up some pictures. All right, we'll start from here. So, sorry. Okay, so you have this group of complement proteins, right? And they get activated, and the activation of one complement protein then activates another one, which activates another one. So that's cascade that I am speaking of. There are three processes within the complement pathway. You have a classical pathway, an alternative pathway, and then you have one that's the lectin pathway. So the proteins that are part of the complement pathway, so proteins get designated with an uppercase C, or they're labeled an uppercase C, right? And then when that main protein fragments, because what happens is you'll have this main protein, it gets activated, it cleaves into two other components. Okay, so those components that main protein has fragmented into are then designated with the lowercase a and b. So you have an uppercase C protein, complement protein C. That complement protein C then is going to get split into C lowercase a and C lowercase b. Okay. So we start off by looking at the classical pathway. The classical pathway starts when an antibody binds to its specific antigen. So let's do some definitions. An antibody, as you should know already, is produced by a B cell when the B cell interacts with its specific pathogen. There is co-stimulation that occurs, and all of this is detailed in my video lectures. So before you start seeing this recording, I urge you to watch my recordings first so you kind of understand what these terminologies are. Then this will make much more sense. Or of course, if you've taken 202, you already should know it. So the antibodies that the B cells produce, the free-floating antibodies in plasma, when they interact with their antigen or the molecule that antibody binds to, what happens then is a complement protein called C1 comes along and binds to that antibody. That C1 that then splits, and then those fragments or those proteins then split also. So let's look at it on the picture. So this is a classical pathway. The complement system has three main pathways, one of which is classical, and that's what we're looking at. So here's a microbe that we're seeing here, right? And it has these proteins or components that are sticking out, right? And then you have these green structures that look like shaped like a Y. They are binding to, these are the antibodies, and they're binding to their specific molecules that they bind to. When that happens, the first thing that's going to happen is you have this complement protein called C1 that comes along and binds to it. It's attracted to bind to it now. Then what happens, right? So the C1 then splits, right? It splits, and it has two major proteins that are formed called C2 and C4. C2 then cleaves once it is formed. It's unstable. It breaks apart into C2 lowercase a and b. And then C4 splits up into lowercase C4 a and b. Now the C2a and the C4b will come together and form C3. C3 then, once it's formed, it's unstable, it breaks apart, and it breaks apart into C3 lowercase a and C3b. What C3a does is it causes inflammation. It causes inflammation because it causes the release of histamine from mast cells and basophil cells. The C3b, what it does is it causes lysis of a cell and causes opsonization. So I thought I had a slide explaining what this is. So opsonization is that the C3 molecule, what it does is it coats the pathogen. It attaches to the outside of the pathogen. And so it's now that pathogen is kind of tagged. Because it has a C3 protein on it, the pathogen is tagged. So when you have other white blood cells there, remember inflammation has occurred, therefore you'll have more white blood cells in that region. Those white blood cells now will be attracted to that microbe because it has a coating of these C3b molecules around it. That's what's called opsonization. Opsonization is when you tag a microbe or you tag something with these molecule so that the white blood cell coming along can detect it and endosynthizes. The C3b also causes the microbe to lyse. It also causes the microbe to lyse. It does that by binding to the cell wall or the outer portion of the microbe, and it kind of forms a hole. That hole is formed, and then of course if there's a hole, water is going to rush in and it causes the lysis of that cell. The second pathway is called the alternative pathway. In this case, the C3 that's present in blood combines with these factors called factor B, D, and P that are present on the microbial surface. And when that happens, the C3 splits into C3A and C3B. And then we're back to here, C3A and C3B. So the only difference is that in the alternative pathway, you don't need this antibody and microbe. It's just, there's an alternative pathway to C3 and C3B. That's why it's called alternative pathway, right? So in this case, the C3 itself binds to these components that are already on the microbe. Now, not all microbes would have this. That's why you have this other pathway, the classical pathway. But the microbe that happens to have these molecules on the surface Well, these are the C3 binds to it. You have cleaving, and then you go through inflammation, lysis of the cells, and opsonization again. Okay. Then the lectin pathway. In this case, what happens is when the macrophage ingests the pathogens, right? When the macrophages go through phagocytosis, remember what I said, you have the release of cytokines. So what cytokines do, and we've already seen two roles for cytokines already. One, the cytokines cause differentiation of the adaptive immune cell. So that cell goes through differentiation. You have more of, proliferation. So you have more of those adaptive cells or more of those immune cells. Second thing the cytokines did is they travel to the hypothalamus and resets the normal body temperature to a higher temperature so that the body will warm up and you'll then have a fever. The third thing that cytokines do is they stimulate the production of lectin from the liver. Now, let's just go to the picture. Okay, so here's the lectin pathway. So here's the microbe. And in this case, if this microbe has these specific components sticking out. So if the microbe has this component called mannose, which is a sugar, if the microbe happens to have mannose sticking out to it, we have proteins, Okay. You have proteins called mannose binding lectin. So lectin, right, that's produced by the liver because of the cytokine, presence of this cytokine. Lectin can bind to mannose, right? And when lectin binds to mannose, it activates proteins C2 and C4. So here's the microbe. Here is lectin. Lectin binds to the mannose that happens to be present on this microbe. When that happens, C2 and C4 are activated, and you notice it's the same thing again, right? Same pathway. So anything past C2, this C2 and C4 getting activated, everything else is the same for the three pathways. So this is how that antibody that's formed by the B cell, what it does. That's free floating, right? It activates the complement pathway, specifically classical. Then you also, because of these complement proteins, you can have either the lectin pathway if the mannose is present, or you can have the alternative pathway. Right? So as I said, that these molecules, specifically C3b, causes lysis of that cell. And it does so by creating a hole. That hole that's created is called a macrophage membrane attack complex, a MAC complex or a MAC. So those proteins, what they do is they kind of like, they form a hole. They form a hole, and so now you have lysis of that cell because water is going to rush in. Opsonization, as I said, it promotes the attachment of phagocytes to that microbe because it's tagging it. It's coding that microbe so that other white blood cells, other phagocytes will have chemotaxi or positive chemotaxi towards that microbe and phagocytize it. Also inflammation, because these complement proteins that have been activated will bind to mast cell, as I was saying, and cause a release of histamine. And of course, we know histamine is a vasodilator. So here is a MAC complex that's formed, or MAC that's formed. Here is the our rod-shaped bacteria, and now they've been they've lysed because this hole that's formed. So this is that MAC complex that I was talking about, right? It forms these holes. Okay. So what are the outcomes of the complement activation? Lysis of the cell, opsonization, and inflammation. Inflammation occurs because the C3A that's formed, so C3A and this, so our C3A directly can activate the mast cell to release histamine. The C3B is going to cause this C5 protein of the complement pathway to split, And then these fragments will also then cause the mast cell or stimulate the mast cell to releasing histamine, which causes inflammation. So this is a bigger picture, right? So here is the mast cell, and it's got lots of these granules that have histamine within them. So this is a C5a that we just learned. It's going to bind to its protein, its receptor, which causes the release of histamine. C3a does the same thing, causes the release of histamine. So we've seen where the C3a comes from. We've not seen where the C5 comes from. So the C5 comes from they get activated by macrophages and neutrophils. And these are going to cause the C5 to split into C5a and B. And the C5a comes along and causes the release of histamine from mast cells. So regulation of the complement proteins. So regulatory proteins will break down the complement proteins once they're formed by the liver, because you don't want to you don't want these proteins to also start destroying self cells. If you lack complement proteins, obviously this is going to leave you susceptible to infections because you don't have this opsonization and the lysis and inflammation process. How can microbes escape these complement proteins? By having a capsule. So capsulated viruses will evade the complement protein. Bacteria have capsules, remember the glycocalyxes, capsules, and the biofilms. So the ones that have capsules can escape the effects of the complement system. When the cell of the body gets infected by a bacteria or a virus, it's going to die, right? Because it's been infected and it's going to die. So what these cells, infected cells, do is they release these chemicals called interferons. So interferons are so cytokines are an umbrella term of chemicals. What's common between these chemicals is that they cause differentiation and proliferation. One such cytokine is an interferon. Excuse me. Interferons are released by cells that are infected. Cells that are infected know that they're going to die, so what they do as a last-minute ditch to save other cells around them, other healthy cells, is they release something called interferons. So interferon alpha and beta, these are produced by cells in response to viral infections. And so what these interferons do, so interfere, right? So what they do is these chemicals travel through paracrine distribution. So they just go to nearby cells. They will then enter those cells, and they cause those cells to produce antiviral proteins, so that these other cells then are prepared for viral infection, right? So they produce antiviral proteins that inhibit viral replication within them. Interferon gamma causes neutrophils and macrophages to kill bacteria. So it activates neutrophils and macrophages. I always forget I have this picture. So this is what I mean. So here's a virus. Here's a virus. It sends in its viral genetic material. In this case, it's a RNA. It will then, this cell is going to die, but what it does before it dies is it produces this interferon. The interferon travels via paracrine to a nearby healthy cell, causes that healthy cell to produce antiviral molecules. In addition to these interferons, you also have these other molecules within the body that also help the immune system. So transferrin regularly is its main function is to transfer iron. So when our red blood cells die, so red blood cells, our red blood cells lose their nucleus, and then they travel through the blood for about 120 days, and after which the cells start dying. And once they start dying, they're brought to the liver where, the red blood cell is taken apart and the proteins are reused, but the iron that's in the hemoglobin, it gets bound to a protein, and then it gets moved from the liver to the bone marrow, where then it can be used again, incorporated into new red blood cells. It gets transferred by being bound to this protein, and it's transferred via this protein called transferrin, right? So what transferrin does is it binds up protein. Lactoferrin is found in milk, saliva, and mucus. Same thing. It is a molecule that binds iron. Ferritin is found within liver, spleen, and bone marrow. It sequesters iron, binds iron, so that because iron is very active, so it prevents iron from being available. Of course, hemoglobin, which is within our red blood cells, that also binds up iron. So you notice that all of these molecules here, they bind iron. So it's going to prevent pathogens that require iron from having access to iron within our body. So then they wouldn't be able to survive. So bacteria to counter these iron binding proteins produce something called cytophores. And what they do is these are molecules that have a affinity for iron, so they compete with these iron binding molecules within our body. So this is the counter that bacteria have in order to get the iron. So these cytophores are going to try to bind the iron and competing with these other iron binding proteins. Now, we also produce antimicrobial peptides. So these are small, amino acid chains that are produced in response to certain proteins and sugar groups that are present on microbes. What these small antimicrobial peptides can do is they can prevent cell wall synthesis. They can form pores, again, forming that hole in the plasma membrane of pathogens. And this is it's called a broad spectrum activity because it's not specific to specific bacteria. All right. So that is the first line of defense. So we go on to now looking at the adaptive immune system. So I thought this was hilarious because here are these bacteria, right? And they're getting getting chased by neutrophils, but they're also getting, shot with these antibodies that are out to get them, right? So that's what we will learn about in the adaptive immune system component. Okay. So the adaptive immune system, unlike the innate immune system, targets specific pathogens, right? Because there are molecules that stick out from the pathogens, and there are the components of the adaptive immune system have a specificity, or they specifically bind to specific components on pathogens. So not all pathogens are, or rather pathogens have specific antibodies or specific B cell, T cells that will recognize them. So adaptive immunity are defenses that target a specific pathogen. It can be acquired either via infection or vaccination. So the adaptive immune system gets activated when it comes across its specific pathogen. So how can it come across its specific pathogen? Either via infection or through a vaccination. And what a vaccination is you have introduced into your body either a piece of this pathogen or its genetic material, just like the mRNA vaccination for COVID that we're just talking about, okay, in the news rather. So there are two responses. There's a primary and then a secondary response. The primary response, of course, primary. So this is the first time that the immune system interacts with that pathogen. The secondary response is when the immune system encounters the same pathogen a second time. The first time when the body interacts with a certain pathogen, it's going to take a while before the adaptive immune system can respond to it, because the adaptive immune system relies upon cells dividing and producing the antibodies, and so that takes time. And so that's why it takes longer to take effect than the innate immune system. But once it's gone through that process and you have the B cells produce the antibodies, there are more B cells present, there are more T cells present. So now, once you've encountered that specific pathogen, your adaptive immune system is ready for it the second, if there is a consequent or second follow-up infection. In that case, this secondary response is much faster. And this is because of that memory, because of the cells that went through mitosis, and you have more of those same cells. So this is a good question. Is vaccination an example of innate or adaptive immunity? Actually, it's not a really good question. I think it's more like, is vaccination activating innate or adaptive immunity? And obviously would be adaptive immunity. So let's take a look at humoral and cellular immunity. So let's look at humoral. So humoral, remember, always means pertaining to body fluids. So humoral consists of components that are within the, well, in the body fluid, so in plasma. When B cells bind to their specific pathogen, and when you watch my videos, you'll see how that happens. When a B cell, so B cells have antibodies that stick out from their cell surface. Those antibodies have a specific pathogen, molecule that it binds to. When it comes across its specific pathogen, the B cell will be stimulated, and it will then attract a T helper cell, and these two are going to interact, and you'll have the production of antibodies. So B cells are lymphocytes because they're produced within the lymphatic system in the bone marrow, red bone marrow. So B cells are lymphocytes that are created and educated or programmed within the red bone marrow. They have these antibodies that stick out from them. And when they come in contact with their pathogen, they interact with the T8 cell and produce antibodies. So they recognize antigen. So by the way, an antigen is something that an immune cell responds to, okay? So the B cells will make antibodies to their specific antigen once it comes in contact with it, right? Don't worry about why it's called a B cell. So this is why B cells are part of the humoral immunity, because they produce antibodies. So I know I'm saying two things. There are antibodies that are on the B cell, and then I'm saying they produce antibodies. So B cells, there are different types of antibodies, and one type of antibody is one that sticks on the outside of the B cell. Okay, that B cell, once it encounters its pathogen with the help of a T helper cell, it will produce lots of the same antibody, but these antibodies are free floating. Remember, we talked about the complement pathway. Those free floating antibodies will activate the classical complement pathway. So these antibodies are in plasma. That's why B cells and antibodies are part of humoral immunity. But then you have where the cells are interacting themselves. That's when you have cellular immunity. This is cell-mediated immunity. This then, this includes, or this involves T cells or T lymphocytes. So T cells recognize antigens. draw this out. So there are two types of T cells. T helper cells. Again, please watch my video, but you should know what T helper cells and cytotoxic T cells. There are two types of T cells, T helper cells and cytotoxic T cells. The way these T cells work is much like the B cell. They have a receptor on them, except their receptor isn't an antibody. Their receptor is called a T cell receptor. Okay, it's a T cell receptor. And what this T cell receptor does is it looks for a specific receptor that's on other white blood cells. The receptor that the T8 cell looks for, okay, so because we are not physiology class, you don't need to know. Okay, it's too much. Okay, so the cellular immunity is immunity that's afforded by cells interacting. And T cells are part of the cellular immunity because T cells interact with other cells in order to have an immune response. That's what this is saying. T cell recognize antigenic peptides processed by phagocytic cells. So whereas the B cell interacts with the antigen directly, the T cell interacts with a piece of an antigen that's been processed by another immune system cell, by another immune cell. Okay, so T cells don't interact with the antigen directly. They interact with an antigen that's been processed by a immune cell. Okay, so that's what this means. It's processed by a phagocytic cell. They are born in the red bone marrow, but they travel to the thymus, and that's where they get programmed. What programming means is where you have the expression of these T cell receptors. Each T cell has its specific T cell receptor. You wouldn't have another T cell that has the same receptor. Okay? So that's where they mature in the thymus. That's why they're called T cells. So the T cells, the T cell receptors that are on the surface of the T cells will, when they come in contact with their specific antigens. Now remember, this is the antigen that's been processed by another cell. And so you have an immune system cell that is expressing this, showing this piece of antigen. And now you have a T cell that comes along and binds to it. And so now you have an immune system cell, a B cell, or phagocyte, and then a T cell, and they interact. When they interact, they co-stimulate each other by releasing cytokines, right? They release cytokines, and then the cytokine causes proliferation and differentiation of that T cell. Again, please watch my video because it's really difficult to explain this in this kind of linear formation. It's better to draw it out and so forth, except it's very hard to draw with my mouse pad. So it's just something that would just annoy you more than teach you anything. But watch my videos because I worked it out on the board. So T cells are part of the cellular immunity because they interact with cells directly. B cells are part of the humoral immunity because they don't interact with the cell directly. They interact via antibodies that they produce. Okay. So this is talking about the difference between B cells and T cells. So you have a stem cell, hematopoiesis occurs within the red bone marrow, and you have the formation of B cells that remain within the bone marrow, they mature there, and then they migrate to lymph tissue. The T cell will be born in the bone marrow, but it travels to the thymus to differentiate or to get programmed, and then they travel to lymphoid tissue, like spleen and the lymph, and then from there enters blood. The cellular immunity, because of the T cells, it can attack antigens that are inside of cells. The humoral immunity can fight pathogens that are still outside of the cell. So T cells recognize infected cells, right? Virally infected cells, some fungus, parasites that might be inside of cells. This is because the cell that's infected, it expresses on its cell surface either damaged protein of itself or pieces of the pathogen that a T cell recognizes and comes along and will have its immune response. The B cell doesn't recognize infected cells. It recognizes a pathogen because of its antibodies. So its antibodies can recognize pieces of a pathogen, and then the antibodies bind to it, and then you have that whole complement pathway that we saw. Okay, so that's how the cellular immunity works versus the humoral immunity. Cellular immunity attacks antigens, or it can recognize infected cells, whereas humoral immunity only recognizes the pathogens, does not recognize infected cells. Okay. So we talked about cytokines for the last lecture, and cytokines are, again, this umbrella term of chemicals that cause cells to proliferate and also differentiate. So cytokines are chemical messengers that are produced in response to a stimulus, and they cause differentiation and proliferation. So the different types of cytokines, we looked at interleukins already. Interleukins are are chemicals that infected cells release, and then these interleukins will travel to nearby healthy cells and cause those cells to produce antiviral molecules. Then you have chemokines, and these are going to cause the migration of leukocytes, so they cause the chemotaxis. Then you have interferons. Oh, I said it differently. Excuse me. I said interleukins and I was thinking interferons. Interferons are the chemicals produced by infected cells. Interleukins are cytokines that are released by leukocytes, hence interleukins, right? Leukocytes. The leukocytes, so remember what I said, when the B cell interacts with its pathogen, it expresses that pathogen or presents its pathogen, and the T helper cell comes along, and then they both release cytokines. When a cell that's dying or it gets infected, it presents its either damaged protein or part of the antigen, and then a cytotoxic T cell comes along and releases cytokines. So these interleukins are cytokines produced by leukocytes to them to interact with each other. Interferons are chemicals released by infected cells. You also have something called a tumor necrosis factor. So tumor necrosis factor is a factor that's released by cells that have come out of their cell cycle, and they're just going through mitosis a lot. So they release this tumor necrosis factor to tell the immune system that I'm going through too much divisions, so to prevent a tumor formation. Okay, so that's a tumor necrosis factor. hematopoietic cytokines. So these are cytokines that control stem cells, right? So that, because remember, the stem cells go through hematopoiesis. So everything is kept under check, right? Checks and balances. And so a hematopoietic cytokine controls that stem cell, so the stem cell doesn't continuously divide. When you have an overproduction of cytokines, that's what leads to something called a cytokine storm. So what do you think could be something that causes an overproduction of cytokines?
Maybe like multiple stimuluses firing off at the same time.
Yeah, or if your immune system is overactivated in a very short time. So a lot of the COVID patients, right, the very quick responses, the very quick, respiratory embolism and so forth that they are experiencing is because of a cytokine storm. So I'm working on a COVID presentation. Just I want to take you guys through just like recent data and recent information. Now, of course, 2 months from now, that information will have changed, but I want us to be on board with what's happening now. So many of the patients earlier, when we didn't know what was happening in March and so forth, is they were coming in with this cytokine storm. And it was just something that you can't control it because it's happening so quickly. And what happens is with the cytokines, it causes massive vasodilation. So that's why you're seeing the pulmonary edema, and they're literally, lungs are just filling with water. And their O2 levels were dropping even if they were on oxygen, because oxygen exchange was just not happening. They're kind of just dying of pneumonia in some ways. So that's the overproduction of cytokines leads to a cytokine storm. So let's go through antigens and antibodies. Okay. So again, an antigen is something that stimulates the immune system, but more specifically, it is a chemical, or not chemical. An antigen is something that causes the production of antibodies. So antibodies are made by B cells. So when a B cell comes across its specific pathogen, it will go through co-stimulation with the T helper cell and produce antibodies. The antibodies then will be free floating, and then when they come across the same pathogen or piece of that pathogen, sticking out from the pathogen, you will activate the complement pathways, right? So these antibodies, so the components that antibodies bind to are called antigens. They're usually on the surfaces of invading microbes, right? Antibodies interact with things called epitopes, or also called antigenic determinants. So the part of the antigen that the antibody binds to is called an epitope or an antigenic determinant. Antigens can either be complete antigens where they cause antibodies to be formed, or they can be hapten antigens. This is where the antigen is, I don't know about too small. Just that the shape of the antigen, whereas an antibody can bind to it, it doesn't really cause an immune response because it's not the right shape. It's not the right shape. So you can have binding and you can stop that antigen from moving around, or antigens attached to the pathogen. So if you can kind of bind it and it can't move around anymore, but it doesn't cause an immune response. You don't have antibodies produced in response to it. So that's when it's called a hapten. So here's a bacterial cell, right? And it has these components sticking out from them. Could be called epitopes or antigenic determinants. And here's the antibody that happens to have a binding site. You could see this V-shaped thing. So here's a, complementary epitope. So this antigen will only bind to these epitopes, right? Whereas this antibody only binds to this shape. So you could see what I mean, that antibodies have specific substrates that they can bind to. So this is what this is showing. These are epitopes. This is what the antibodies are going to bind to. And then you can put, once that happens, then you activate the complement pathway. Classical in this case because it involves antibodies. So hapten molecules are molecules that can, you can skip slide 17. Just know that hapten molecules don't elicit an immune response. Okay? You don't have to know about carrier molecules in this. So antibodies. Antibodies are molecules that are produced by B cells and released into plasma. They are actually called globular, or they're actually called immunoglobulins because they are immune proteins. They're large proteins, and so they're globulin, and they're called immunoglobulins because they're part of the immune system and produced by immune systems. You don't need to know about valence. You don't need to know about the different parts of the antibody. What you do need to know that there are 5 classes of immunoglobulins. Immunoglobulins are written as I lowercase g. And the way I remember it is the name MAG, right? So you have IgMs, IgAs, IgDs, IgGs, and IgEs. You don't need to know this. Let's go through what each one of these are. So IgMs are made of 5 different antibodies held together. Okay, you don't need to know the J chain and the pentamer. Just know that they're made up of 5 monomers. They make up about 6% of, serum antibodies. These remain within the blood. What they do is they cause well, it says clumping of cells. I can't think of another way to say it. What they will do is they bind to their antigen and they kind of sequester the if it's a cell or if it's a virus that it binds to. If the bacteria or the virus has this specific epitope sticking out that the IgM binds to, it will sequester these things and cause clumping of them and causes precipitation of that. Usually it's a first response to an infection, and IgMs are very short-lived. IgMs make up about 13% of serum antibodies. These are present in mucous membranes, saliva, tears. Right, earlier I was talking about having antibodies in tears, so this is what these are. IgAs are present in these fluids that are present in places where we interact with the external environment, and it prevents microbial colonization and attachment to mucous membranes. IgDs are similar to IgGs, which we'll see in a minute. They can be in blood or lymph. These are the antibodies that are on B cells. Remember, I was talking about B cells, and they have antibodies sticking out. The class of antibody that's sticking out is the IgDs. Okay, they basically just help bind the specific antigen, and then the B cell gets activated, and then you have more antibodies that are formed that are now free floating. IgGs, these are present in blood, lymph, and also within the intestines. They make up most of the serum antibodies, about 80%, of course. IgGs can cross the placenta, so the type of IgGs that mom has can be passed on to the baby, the fetus. IgGs trigger complement pathways. They therefore enhance phagocytosis because complement pathways involves opsonization, neutralizes toxins and virus, and then of course protects the fetus because it can cross the placenta. IgE. So IgEs are antibodies that are going to that are present on mast cells and basophils. They can also be free floating in the blood. So when they're formed, they are going to be attracted to mast cells and basophil cells. Mast cells and basophil cells are immune cells that have histamine. And when these cells are activated, because when the IgE gets activated, it causes the release of these vesicles, so you have histamine present. So histamine causes vasodilation. So that's why IgEs are involved in allergic reactions, right? So it causes the release of histamines. So when the IgE antibody that's present on the mast cell and the basophil binds, of course, it's an antibody. When it binds to an antigen, it causes the release of histamine. It also causes the lysis of. So IgEs have dual functions. Pictures say 1,000 words. Do I have pictures? Oh, no, it doesn't. Okay. So here's the IgE. IgEs are part of allergic reactions because they're present on mast cells and they're present on basophil cells. When the IgE is activated on the mast cell and the basophil cells by complement proteins, remember complement proteins cause inflammation, that's when the vesicles get released with histamine and you have vasodilation, and this is part of the allergic reaction. IgEs are also present on eosinophils. So when eosinophils come across a parasitic worm or a larvae, that then causes the release of the hydrolytic enzymes from the eosinophils, and therefore it causes the death of parasitic worms. And that's how IgEs work. Okay, so they're bound to mast cells and basophil cells, and also when they're, ooh, my neighbor got hurt. That's not funny, but anyway. Eosinophils, and they will then, eosinophils release hydrolytic enzymes, and of course they are going to destroy worm larvae that are present, that can be present. So let's talk about now, okay. Let's move along and learn about humoral immunity response processes. So humoral immunity involves a role, or involves antibodies. So antibodies are produced by B cells. When a B cell, which has its antibodies sticking out from it, interacts with a specific antigen, that B cell then is going to present a piece of that antigen on its cell surface. When it does that, a T cell, specifically a T8 cell, comes along, recognizes that, and if it happens to have the T cell receptor that recognizes that epitope, that antigen piece, it'll bind to that B cell. You have co-stimulation, and then you have more of those T8 cells, and you have more of those B cells, but you also have more of that same antibody, and now that's free floating. So what we're going to learn here is compare and contrast T dependent and T independent antigens. So let's see what that is. Okay. When we're talking about T cells, I said that there are two types, TH cell, right? And then the other, that's T helper, and the other is cytotoxic T cell. These two T cells, they're cell-mediated immunity. T cells bind to receptors that are on either the self cell or on other immune cells. T8 cells look for and can bind to proteins called class 2 MHC receptors. These are present on macrophages that present antigens. They are called APC cells. Please watch my videos because I explain everything from the very, I explain everything out clearly in those videos. Right now, I can't explain to you what an APC cell is because you should know what an APC cell is already. Okay, so T8 cells bind to, so let me just take a little poll. Did you guys learn all this in your immune lecture for in your 202?
I haven't even taken 202, Professor. So
Yeah.
myself, no.
Oh, I thought everyone here had already taken 202. My bad. Okay, Brian, Kim, you guys there?
But I will watch the videos.
Yeah, the videos. It's my 202 lecture, and it takes you through this step by step.
Yes, we did go over it in 202.
Okay, so you guys know what T8 cell and TC cells are, the MHC 1 and 2.
Yep. And then I already watched the videos from.
Oh. Okay, were they helpful?
They were, yes.
Okay, great. Yay. Okay, so all right, so then I don't have to be so concerned. So we already know this, right? So T8 cells, well, one, you will know it when you go watch it. T8 cells bind to class 2 MHC, and TC cells bind to class 1 MHCs. Right. I always forget that I change this. So B cells, when they come in contact with their antigen, they phagocytize that antigen, and then they display that antigen on their cell surface on their MHC class 2 molecules. And then a T8 cell will come along and bind to that. If that T8 cell happens to have a T cell receptor that recognizes that antigen piece, when that binding occurs, you have co-stimulation. And what will happen is the T helper cell goes through differentiation and proliferation, and you form memory cells and you form effector cells. The difference between memory and effector cells is that the memory cells live longer than effector cells. And when a memory cell comes in contact with that same antigen, it then stimulates into an effector cell, and the effector cell is then the cell that actually causes the effect, hence called the effector cells, right? So the T helper cell that has bound to that B cell via the the T cell receptor, you have this co-stimulation because each cell releases a cytokine. Then you have co-stimulation, and the B cell goes through, multiplies and produces memory cells and plasma cells. The plasma cells will produce lots of those same antibodies that are now in plasma. And of course, the T8 cell goes through proliferation and differentiation. So the B cell undergoes proliferation, which is going through divisions. Okay, so here's the B cell. It's come in contact with this antigen. It brings in the antigen and breaks it apart. Then it expresses that piece of that antigen on its MHC class 2 receptor. T8 cell comes along. If it happens to have the complementary shaped T cell receptor, it'll bind to it, and you have co-stimulation, release of these cytokines. And what will happen is the B cell will grow, will go through divisions and producing memory cells and plasma cells. So this is a plasma cell, and you know that because it's producing and releasing the same antibody. Then these antibodies are free floating in plasma. If they come across the same antigen again, then it'll bind to it, and then it will cause the formation of that cascade of complementary pathway. So this differentiation of those B cells goes through a process called clonal selection. So clonal selection is the selection of the proper type of B cell. So that B cell goes through divisions, and if the proper type of antibody is not produced, then those B cells that don't have the proper antibody go through a clonal deletion. So it eliminates harmful B cells. But clonal selection is selection of the B cells that have formed from that division of the proper antibody producing B cell. So this is the whole process that I was just talking about, nicely illustrated. So T-dependent antigen and T-independent antigens. So T-dependent antigen is an antigen that requires a T8 cell in order for an antibody to be produced. So what we just looked at with the B cell, with the antibody and binding the antigen, causing then T8 cell binding and co-stimulation, and then more B cells, and then antibody production. So that is clearly T-dependent antigen. You can also have antigens that stimulate B cells to produce antibodies without the help of T helper cells. Those are called T-independent antigens. This is, the difference is these promote, you have a weak immune response because you don't have T8 cells also that went through proliferation, right? And it produces IgMs. You don't have memory cells. So the B cell doesn't go through proliferation. It just produces more of those antibodies. So usually just produces IgMs, and there's no memory cells produced in that process, right? So in this case, this is an antigen here, and these antibodies that are on the B cell, they're binding to it, and then this B cell, because of this specific type of antigen, it'll produce more of these antibodies and release them, but the B cell doesn't need a T8 cell to bind to it, doesn't need co-stimulation, and therefore it itself doesn't go through divisions, right? So you have a very weak immune response. All you really have is more antibodies produced and released. So let's look at the outcomes of an antigen antibody reaction, right? So we're talking about antibodies that are formed. Now they're free floating. Well, what happens when that antibody, free floating antibody, it's not attached to a cell? What happens when it comes in contact with this antigen? So picture is always better. Right? So in this case, here's an antibody, and it's bound to these bacteria here, right? So the different things that can happen when an antibody binds to its specific antigen are agglutination, opsonization, antibody-dependent cell-mediated cytotoxicity, neutralization, and activation of the complement system. So agglutination is when you have blood clot forming. So agglutination occurs when you get blood that's different from your blood type. So blood type is our red blood cells have on their cell surface protein markers. Either they have protein markers sticking out or you don't. If you don't have any protein markers sticking out on from the red blood cell, that's called a type O. If you have a type A protein sticking out, then you're type A. If you have type B protein sticking out, you're type B. You can have both, in which case you're type AB. So the body will have antibodies that are looking for the type of protein that you don't have, right? So if you get a blood type or blood type that's not the same as the blood type you have, then you can have these antibodies that bind these different red blood cells up, and you will have blood clotting formation. So that's agglutination. Opsonization, we've already seen. This is where the antibody will bind to its specific antigen that's on a pathogen, and it coats it. So that's opsonization. Antibody-dependent cell-mediated cytotoxicity. So this is where you are coating these bacteria. So this is opsonization. This is where you cause the lysis. So remember the complement pathway. Remember it causes the C3b causes the MAC complex to form, and you can cause lysing. This is where you have this pathogen. Excuse me, this is a neutrophil. So this is where this pathogen, this bacteria, has been coated with those bacteria, with these antibodies. And so this is opsonization. This is actually sequestering the bacteria or virus and preventing it from moving around, so you keep it in one place. In this case here, what you're showing here is so this is a parasite. It's got these epitopes, and these antibodies are binding to this epitopes. And in this case, it is then bringing over the eosinophil, because this is a parasite. So this is like a helmet or worm or larvae. And this eosinophil is going to come along and then release the enzymes and cause lysis of that parasite or death. In this case, this is a virus, and this is a virus in here, or this is a bacteria, and these antibodies have coated these microbes and prevent them from moving around, right? So in this case, you've neutralized these bacteria. In this case, it's showing a toxin that's been released by a bacteria or even a viral protein, and these antibodies have bound it all up, thereby detoxifying that toxin. Okay? So neutralization. Okay. So let's talk about cellular immunity. Cellular immunity is all about T cells. So T cells, born in the red bone marrow, they travel to the thymus, where they go through a process of selection and rejection. If the T cell is, so T cells travel to the thymus, and they're tested for their effectiveness. If a T cell binds too tightly to self cells, they get killed off. It's such a specific process. About 95% of T cells that go to the thymus to get educated get rejected and killed. You have to be really specific, because cytotoxic T cells can cause our cells to kill themselves. And we'll go through that process. So within the thymus, they go through these T cells that are born in the red bone marrow, go through a thymic selection. So where you have a selection of T cells that have the proper type of T cell receptors that's not going to bind to your self cells and only to, well, non-self cells. T cells that bind to our self cells at all get eliminated, right? So they get eliminated via this thymic selection. When these T, so what this maturing in the thymus means, this is where the T cell expresses its T cell receptor. And then that T cell receptor is tested. If it binds to self cells, that T cell is gotten rid of. Okay. So when a pathogen enters, you don't need to know slide 43. We can skip 43. Okay. Let's talk about cells that T cells interact with. The cells that T cells interact with depends on the T cell. You have a Th cell, T helper cell, and then you have a cytotoxic T cell. T helper cells interact with cells that are called antigen presenting cells, or APCs. Antigen presenting cells are cells that do that. They present the antigen. So antigen presenting cells are all our macrophages. Macrophages bring in the pathogen, break it apart, and then they release the pieces of the antigen, right? Which then goes through blood and gets released from the body. It also holds on to pieces of the antigen and presents that pathogen on its self surface, or piece of that antigen on its self surface. It presents that pathogen, excuse me, I keep saying pathogen. It presents the piece of that antigen on its MHC class 2 complex. So APC cells have MHC class 2 receptors. And so TH cells, or T helper cells, only recognize MHC class 2 molecules or receptors. And then they will bind if that antigen that's on that is complementary to, it can bind to it via its T cell receptor. So macrophages, as I said, are APC cells. They get activated by cytokines that could be present or by phagocytosis. They migrate to the lymph tissue where they present that antigen to TH cells. Dendritic cells are also engulf and degrade microbes, so they're also phagocytic. And then they also will present that antigen on their MHC class 2 to TH cells. Dendritic cells are found within the skin, the genital tracts, within lymph nodes, which is spleen, thymus, and also in blood. Okay. This is a great picture of showing this. These are T cells here that are interacting with parts of this pathogen that you see here. This is showing, so this is a inactive macrophage, and this is an activated macrophage. And you can see these foldings that are present now, right? So that's how you know that this is an activated macrophage. So there are two classes of T cells, which I've been talking about all along, a TH cell and a TC cell. TH cell is T helper, TC is cytotoxic T cells. The TH cells sometimes are also called CD+4 cells because they have an abundance of this protein called CD4. The cytotoxic T cells are called CD+8 cells because they have an abundance of the CD+8 proteins. The TH cells bind to MHC class 2 molecules that are present on B cells and APC cells. The cytotoxic T cells bind to molecules to MHC class 1 molecules. MHC class 1 molecules are present on all of our cells except for red blood cells. So I'll say that again. The MHC class 1 molecules are present on all of our cells except for our red blood cells, because our red blood cells have the type A or type B proteins, or both of them sticking out for them. And also we have red blood cells that don't have any, right? But red blood cells do not have MHC class 1 or 2 molecules. MHC class 2 only present on B cells and APC cells. MHC class 2 present on all cells. When I say that they're present on all cells except for red blood cells, would APC cells and B cells also have class 1 molecules, MHC class 1 molecules? Present?
Yes.
Yeah, absolutely, right? So when our body cells get infected, they are going to present a piece of that viral protein or bacterial protein on the MHC class 1 molecule. The CD+8 or the cytotoxic T cell will come along and bind to that antigen. And when it does, it causes that infected cell to kill itself. The process of suicide of our cells is called apoptosis. So we'll come to what apoptosis is. We'll talk about our CD4 cells first. So the T cell receptors on the T8 cells recognize and bind to MHC class 2 molecules only. MHC class 2 is present on APC cells and B cells, which is also in APC cells, right? When they bind, when you have binding of the T8 cell to the B cell or the APC cell, you have this co-stimulation and you have the release of cytokines, and then of course the proliferation of that T8 cell. T8 cells produce cytokines, and when they are activated through the co-stimulation, depending on the kind of cytokine that's released by the other cell, that T8 cell is going to can differentiate into different types of T cells. So you have Th1 cells, Th2, Th17, and then of course memory cells. So let's see what these are. So this is the interaction. So here's our APC cell, it takes in that antigen, goes through phagocytosis, and now it's presenting a piece of this antigen on its MHC class 2. Here comes along a T8 cell that happens to have a T cell receptor that can bind that antigen. You have co-stimulation, so cytokines are present, and then you have an activated T cell, and then that activated T cell goes through differentiation and proliferation. The cytokines cause a T8 cell, right? So these cytokines cause a T8 cell to go through division and also, which is proliferation. So proliferation and differentiation. So this T8 cell can differentiate into different T8 cells. Now, Th17 cells produce interleukin 17. Interleukin 17 is a cytokine and it can cause inflammation. Th1 cells produce interferons gamma. So interferon, remember, so activates macrophages, enhances complement pathway, can stimulate antibody production. Th2 cells activate B cells to produce the IgE. IgEs activate eosinophils, hence IgE. That's why it's called IgE. IgEs activate eosinophils, but IgEs also cause degranulation of mast cells, right? So sometimes when you have larval infections, you also have inflammation involved, and this is because IgE, right, is present. So this is a good slide to study off. This is exactly everything that I've said, but in illustrated. Then you have T regulatory cells. So this is not something that's in the in my recordings because that was for 202s, and this is, you know, the next step in that information. So there's a subset of T8 cells or CD4 plus cells that carry an additional molecule along with the CD plus 4 called the CD25 molecule. And this is a regulatory molecule, right? So this molecule, when it's present on the T8 cell, that T8 cell is now a T regulatory cell because of that regulatory molecule. It suppresses T cells against, so it prevents the T cell from being targeted by other T8 cells or even itself against other T8 cells. It protects the intestinal bacteria. So we should have asked throughout this whole lecture, we have so many microbes in our intestine. How come our immune system isn't killing them? They're foreign cells. They're not going to have the same markers on them. I mean, they should be getting killed. But why don't they? They don't because they protect these T regulatory cells, protect the intestinal bacteria, right? Also protects the fetus. So it prevents the body from attacking these clearly foreign organisms within the body. Then you have cytotoxic T cells, which I talked about earlier. Cytotoxic T cells bind to MHC class 1 molecules. MHC class 1 molecules are expressed by all of our cells except for red blood cells. So when our cells are infected, what they're going to do is they're going to present on the MHC class 1 molecule a piece of the virus or viral protein or a piece of bacterial protein, something to let the TC cell know, hey, I've been infected. So when that happens, the TC cell, if it has a complementary T cell receptor, same thing like we saw with the TH and the APC cell, you have binding and you have co-stimulation, right? So activations of the cytotoxic T cell, also called CTLs, with the help of T8 cells and more specifically co-stimulatory cytokines that are released, they will cause the cell that's infected, that it's binding to go through a process called apoptosis, and that's called controlled or regulated suicide, right? So the CTL, once it binds to the cell, releases perforin and granzymes. These are molecules that cause a cell to go into apoptosis. So this is, you know, a normal cell, a cell that's infected. It interacts with the TC cell. TC cell then causes the cell to go through self-destruction or controlled suicide. Now, so cells can die via two methods. One is called necrosis, where the cell lyses. The other is apoptosis, where the cell doesn't lyse, the cell implodes. It implodes, it shrinks up, it shrivels up. Everything, all the you know, internal parts of the cell also shrivels up. It shrivels up, and then it's just this, you know, shriveled cell. Of course, it's dead now, and the macrophage can come along and get rid of it. Now, why do you think that this apoptosis is incredibly expensive? There are many enzymes involved. There's a lot of energy involved in sequestering those enzymes and so forth. So why not just let the cell die? It's dying anyway. It's dying anyway. Why have this very expensive process? Forget, in addition to the cost of the enzymes, right? You still have to now have the cost of having these T cells and the proper receptors and so forth. Why is this important? Why did this evolve? Why not just let the cell die? It's dying anyway.
Wouldn't it be to kind of sacrifice the one to protect the many in the body, depending on what it is?
So you're right, but how are you protecting the many? It's only the cell that's infected that's going to die. Or is it?
Well, like in our last module, at least with the viruses, it released
That's true.
the
The
what are
viral.
they called? The viral DNA to infect more cells, so to prevent that.
Right. And also if the cell lyses, A, can spread the pathogen. B, the cell has lots of these lysosomes, remember, and you have a lot of hydrolytic enzymes and things like that. So you can damage other cells around you. So necrosis is cell death, but through cell lysis, and that can be damaging to other cells. But to counter that, to prevent the cell from doing that and prevent further pathogen spread, as you said correctly, Kim, this is why the evolution of apoptosis, which is a very expensive process, but it is a way to keep the pathogens from spreading, whatever agent that's killing that cell from spreading. So again, apoptosis programs cell death, prevents the spread of this infectious agent, such as a virus or bacteria and its toxin. And so what happens is you go through this programmed cell death where the cell just implodes. So here's a cell, and this is a cell that's going through apoptosis. It starts imploding. Now we talk about cells that are called natural killer cells. So natural killer cells are granular leukocytes. These cells can destroy cells without the necessity of binding to an MHC class 1 like the TC cells have to have. They can kill viral infected cells and also tumor cells, and they can also attack parasites. They don't require binding of a specific antigen, but what natural killer cells detect on cell surfaces are different glycoproteins or glycolipid proteins that are sticking out. So they're not really looking for antigens. They're looking for specific types of, I won't say, they detect not of the self, so they detect abnormal glycolipid expression on cell surface. What they do is they cause pores to form on the target cell, causing lysis or even apoptosis. Okay. So great slide to study off of, gives you everything in a nice table. Antibody dependent. So let's talk about how protozoans or worms, helminths or worms, get killed. So these are too large to be phagocytized, as I said earlier. And so what happens is you're going to have cynophils that are going to come along and destroy these cells. So they have protozoans and larvae, worm larvae can get go through a process of opsonization where the antibodies come along and bind to the surface. So that's going to then tell immune cells that to come and attack it. So what happens is protozoans and helminths get coated with antibodies. The immune system cells attach to regions on the antibodies, and then now the target cell gets killed by eosinophils that come along and release hydrolytic enzymes on them, right? So what you see here is our large parasite, and you have these antibodies that bind to them. And what's going to happen, there are regions on the antibodies that are recognized by macrophages, eosinophils, and they will come along and release hydrolytic enzymes, destroying that this parasite. So this is a picture, or excuse me, scanning electron microscope picture here of a fluke, which is a type of helminth or type of worm. And these are all these eosinophils around it. killing it by digesting it from the outside. So immunological memory. So remember what we said all along, right? APC cells, when they interact with T8 cells, you have co-stimulation, and the T8 cell goes through proliferation differentiation, and that creates memory, right? The proliferation at first produces two different groups of T8 cells, and memory cells and effector cells. The memory cells hold the memory of that antigen, so you have now memory, right? So that's why the first response takes a while before something can happen, because you have to go through this process, you have to go through a proliferation differentiation, and then the production of more of those cells. And if we're talking about B cells, the production of antibodies. So that's why the secondary exposure to the same antigen is going to be faster than the primary exposure. So secondary responses occur after the second exposure to the antigen. You have a much rapid response, you know, and of course the symptoms are alleviated quicker, right? We can check to see if somebody was exposed to a certain type of a antigen by performing an antibody titer. So this is just looking at the amount of that specific antibody that's present, right? So it can show you the intensity of that humoral response. It can tell you how badly you were, or what was the magnitude of that infection, right? Because the magnitude of the infection is going to give you, the more antibodies you have, the more particles, pathogenic particles you had. The first antibody that gets produced is the IgM. The secondary exposure produces the IgGs. And this is what this is showing. The initial response is IgMs. Then when you have a secondary exposure, the type of antibodies that are formed are IgGs. Now, you can get this adaptive immunity. Remember, adaptive immunity is the presence of these antibodies. So there are different ways you can get, you can acquire adaptive immunity. So one is, of course, naturally acquired active immunity. This is when your body has been infected, and now your B cells are responding to it, and of course, that's how you then have those antibodies. You can have a naturally acquired passive immunity. So this is when antibodies are, you're acquiring the antibodies, either transplacental or via, so naturally acquired, right? So it's not that you've been given it exogenously. Then you have artificially acquired active immunity. So this is vaccination. But you're getting, what's the difference between artificial acquired active and passive immunity? Artificial acquired active immunity is where you're activating the immune system. You are getting an injection of a piece of a viral particle that's been, you know, it's been changed so that its instructions to take over a cell is removed, or a piece of the virus. So like with the mRNA that's being used in the vaccinations for COVID. So your immune system is exposed to this antigen, and then it can go through this whole process of forming antibodies, and now your immune system is ready for an active form of that pathogen. Artificially acquired passive immunity is when you are just getting the antibodies. Now, in this case, this is not long lasting because you don't form any memory cells, or you don't have a memory. When you have artificial acquired active immunity, you have memory because you have memory cells that are formed. When you have artificially acquired passive immunity, you're just getting the antibodies. And so you don't get it. So also, we were talking about naturally acquired passive immunity. So it could be transplacental, or it could be through milk, right? Mother's milk. Okay, so again, great slide gives you a good flow chart of everything that we just talked about. Let's see if I really need you to know that. Okay, you can skip 75 and 78. And so this is a great slide because it is, it gives you a good snapshot of, let me move this out of the way, of the differences between humoral and cellular or cell-mediated immune systems, right? So this is everything that we've talked about, actually. So this is a good summary slide. Okay. That's the end of that. I will stop recording and I'll post it as soon as